Your body makes over 100,000 proteins. But do you know how it makes just one of these proteins? A protein is made up of a chain of amino acids. The directions for building these amino acid chains are found in sections of DNA called genes. So how does your body decipher DNA and use its instructions to build an amino acid chain? It uses a process called protein synthesis. Scientists discovered that protein synthesis is a two-stage process, where DNA is transcribed to messenger RNA, and messenger RNA is translated into an amino acid chain. The first stage of the process begins with a section of the DNA called a gene, which is found in the nucleus of a cell. Yet the instructions for protein building are needed in a cell's cytoplasm. So how do the instructions get from the nucleus to the cytoplasm? Well, in a process called transcription, a copy of the DNA is made, and this copy is able to leave the nucleus and travel into the cytoplasm. There's more than just DNA involved in transcription. To make a copy of a gene, the following molecules are needed: the enzyme, RNA polymerase, and RNA nucleotides: adenine, cytosine, guanine, and uracil. Let's take a detailed look at transcription. We'll look at how the body makes an important protein called insulin. Insulin plays a role in controlling blood glucose levels by binding to specific receptors on a cell's surface, and in doing so, making the cell membrane more permeable to glucose. The gene that provides the instructions for building the protein insulin lies on chromosome 11. Transcription starts when the enzyme RNA polymerase binds to an area at the start of the gene called the promoter region. This enzyme untwists a section of DNA, opening up a gap between its strands. Once there's a gap between the strands, a copy can be built. The copy is different from DNA because it's built out of RNA nucleotides instead of DNA nucleotides. The copy that's built of RNA nucleotides is called messenger RNA. 3 of the 4 RNA nucleotides are the same as those of DNA: adenine, cytosine, and guanine. And one is different: uracil. Pairing of the nucleotides is essentially the same as in DNA, but in the messenger RNA copy, uracil replaces DNA's thymine. The copying or transcribing of DNA into messenger RNA continues until the enzyme RNA polymerase reaches a termination sequence, signalling the entire gene has been copied. The end result of transcription is a copy of the gene in the form of messenger RNA. And it's this copy that leaves the nucleus and travels into the cytoplasm to begin the next stage of protein synthesis: translation. To make an amino acid chain from a messenger RNA strand, the following molecules are needed: an organelle called a ribosome, and transfer RNA molecules. Translation begins when the ribosome binds to the start codon on the messenger RNA. The ribosome reads the nucleotides in groups of 3, called codons. The start codon is a universal code in all multicellular animals: A, U, G. Transfer RNA molecules have a complementary sequence of 3 nucleotides called an anticodon. The transfer RNA molecules attach to the messenger RNA. Each transfer RNA carries an amino acid that corresponds to the codon of the messenger RNA. The first amino acid in the chain is methionine, coded for by AUG. As the messenger RNA is read and transfer RNA molecules deliver the corresponding amino acid, a growing chain is produced. The chain continues to grow until the ribosome reaches a stop codon, prompting the ribosome to separate from the messenger RNA, releasing the amino acid chain into the cytoplasm. Like any amino acid chain, it's not a functional protein until it folds into its 3-dimensional shape. So protein synthesis is a 2-stage process: transcription, then translation. Transcription builds a copy of the gene, messenger RNA, that leaves the nucleus and travels into the cytoplasm. Once in the cytoplasm, this copy is read and translated into a chain of amino acids, which fold into a functional protein.